students, welcome to this lecture of Applied Environmental Microbiology, where we will be focusing on built environment. The built environment in itself consists of everything that has been built, and thus is very any civil infrastructure that has been built, and thus it is very diverse in its scope and involves very different kinds of microbiological processes that happen in built environments. For example, perhaps the room that you are sitting in, the room that we eat, and in the room in which I am recording, all of them include built environment, and because of their different distinct characteristics, and local environment will have very different microbiology. Built environment also includes things such as wastewater treatment, water treatment, because we have basically built them. It also includes the pipes and the buildings that we create, and since all of them are included in built environment, we will first start with advanced treatment in wastewater treatment, and then move on to what happens in our concrete pipes, and have already talked about corrosion in water distribution system. So that is also a typical example of microbiology in the built environment. So let us get on with the built microbiology. Now in wastewater treatment plant, a typical primary treatment process will remove 50% of BOD, will get rid of lot of COD, will get rid of lot of suspended solids and the big material. Then the secondary treatment will get rid of most of the BOD, COD and nutrients. However, at times the end product of secondary treatment, which is biochemical degradation of waste in wastewater, is not sufficient for us to meet the standards. And also at times we might now become aware of contaminants that our wastewater treatment plant was not actually built for. In this case, we need to add a tertiary treatment step or advanced wastewater treatment steps. For example, nitrogen and phosphorus are typical nutrients that we want to remove and are not effectively removed in the secondary treatment process, and to remove them we often need to add advanced wastewater treatment steps. Now nitrogen and phosphorus, if you recollect, are very important because they contribute to algal growth and algal blooms in our lakes and result in eutrophication, accelerated eutrophication of the lakes. So secondary treatment is often not sufficient for achieving desired standards. Now this is your typical secondary treatment process. So therefore we include the advanced wastewater treatment, which includes tertiary treatment, physico-chemical treatment, or combined biological and physical treatment. For example, if I want to remove phosphorus, I have two approaches. I can either have physical removal or purely biological, or I can combine them. Thus all of them would still be my advanced wastewater treatment process. Typically they are used to remove organic matter that is still not removed from our secondary treatment process, suspended solids, nitrogen, phosphorus, dissolved organic carbon and toxic materials. Interestingly, the toxic materials might include, and often do include, emerging contaminants. So the thing with emerging contaminants is that they are persistently released into the environment, and because they are always continually being released in the environment, their longevity does not really make a difference. How long they persist in the environment does not make a difference, because there is always a continuous influx of emerging contaminants. Now here is the thing, only 20% of phosphorus that comes into the influent wastewater treatment plant is actually removed from primary and secondary treatment. So we still have plenty of phosphorus that we need to get rid of before disposing our treated wastewater into environment. Now typically the chemical processes involve coagulation, flocculation using aluminum or iron. Often iron is used, and iron precipitate of phosphates and other phosphorus based compounds results in removal of phosphorus. However, there is a major limitation of this, which is that chemical precipitation of phosphorus makes up to 95% more sludge than simple secondary treatment would. So this, and sludge processing, sludge removal, sludge management is quite an expensive affair for any wastewater treatment plant. So definitely we do not want to double the volume of sludge. If there were an easier way to get rid of the phosphorus without creating more sludge, it would be very preferable, and there to our rescue comes enhanced biological phosphorus removal. So here we have EBPR, enhanced biological phosphorus removal. Now the way it works is that we have sequential passage of our water that has been treated initially through first anaerobic chamber and then through an anoxic or aerobic chamber. Now, this is what your typical ABPI process would look. You have first passing it through an anaerobic chamber and then through an anaerobic or anoxic chamber, and then you have a clarifier which is secondary settlement tank, and then a part of it is returned activated sludge and a part of the it is wasted, and then there is effluent that you send for disinfection. So, there is a disinfection step here, and then you dispose it in the environment. Now, in the anaerobic reactor, what happens is that the phosphorus is accumulated by phosphorus accumulating organisms. So, they are shortened as PAO. So, there are a lot of PAOs present here. What they do is they use the energy that is present in polyphosphate in the water, and they assimilate short chain fatty acids. So, in the anaerobic chamber, short chain fatty acids are being consumed. So, their energy that is stored in this short chain of fatty acid is being consumed, and the next step is the microbes, which are PAOs. They store the phosphorus inside their cell. So, this is intracellular. So, inside the cell they store in form of polyhydroxyalkanates, and they are often written as PHAS. So, we have polyphosphate lying outside in wastewater. Now, these PAOs, phosphate accumulating microbes, they will burn, they have lot of short chain fatty acids in them, they will burn the energy and they will use this energy to accumulate them polyphosphates outside, extracellular polyphosphates, and make intracellular PHAs. So, now the phosphate has produced intracellular PHAs. In the next step after anaerobic treatment comes the aerobic phase, where now the PHAs that has been stored is burnt. So, now the microbes are rich in PHAs, and these are your PAOs, by the way, phosphate accumulating organisms. They are rich in PHAs, but they are low in their short chain fatty acid. So, they burn the PHAs, and they give, when they burn, they release lot of energy. So, they have, and carbon, so they have lot of carbon and they have lot of energy. To increase their biomass. Now, when this has been done, what they do is they convert the available phosphorus, into intracellular polyphosphate. So, remember initially we had extracellular polyphosphate, now we have intracellular polyphosphate, and thus the phosphorus that was lying outside, the extracellular polyphosphate, is now inside the microbe. This is your PAO, by the way. I have made the PAO rectangular in this case, but yeah. So, now what happens is that the biomass that has been produced using this energy and by burning PHAs and converting it into intracellular polyphosphate is, if it makes sludge, and this sludge is removed, and now you have removed your phosphorus. So, the basic principle is the polyphosphate that is outside the cell. So, if your cell is circular again and you have polyphosphate outside the cell, it is taken in, converted into PHAs, and then in the aerobic zone the microbes convert this into intracellular polyphosphate, and now you can remove the biomass, and thus you have removed the phosphate present in the wastewater. So, a typical ABPI process would look like this. The total PHAs would first increase in the anaerobic zone, and then in the aerobic zone, as they are getting degraded and intracellular polyphosphate is being accumulated, they would decrease, and in the settling they would remain constant. Now, your COD in aerated zone will act in the anaerobic zone, it will drop because the short chain fatty acids are being consumed, and then it will not rise again. Your polyphosphate in the anaerobic zone would increase, and then in the aerobic zone, this is your extracellular This is your intracellular. Because it is intracellular, in the water it decreases. Now the cool thing that you need to notice here is that with time how things would change in the typical ABPI process. The short chain fatty acid would do a quick decline. So this is your short chain fatty acids. Your extracellular polyphosphate would first rise and then it would decline, and then your intracellular polyphosphate would rise and then decline. So this is extracellular, this is intracellular, and then the intracellular polyphosphate would rise, and it first stay low and then it will rise, and then this is what is removed as your biomass. Alrighty. Now there are some limitations with ABPI process. One of them is that it fails when it is competing with other microbes. So if we have lot of energy release when the pHs are converted into polyphosphate, and then this is your extracellular polyphosphate, and then microbes will assimilate them in the first anaerobic process, and then your intracellular pHs would increase, and then in the aerobic zone they will decline too. And what will happen is that your intracellular polyphosphate, once the pHs start degrading, they would increase, and the phosphate that you had as polyphosphate extracellularly would be stored here as intracellular polyphosphate. Okay. There is a major limitation with ABPI process, which is that it fails when the competing microbes take over. So in this case, when the pHs are degrading, lot of energy is being produced, lot of carbon is being released, so biomass is increasing, and if instead of our PAOs, which are phosphate accumulating organisms, other microbes increase, then they will be very easily outcompeted. They are not very quick to thrive in diverse competing microbial communities. And they are particularly outcompeted by microbes that store glycogen. So the carbon and energy they have released will be used by these glycogen storing microbes, and the EBPR key microbes, PAOs, will be outcompeted. The important thing about this PAOs is phosphate accumulating organisms is that their ecology and their microbial community structure, their functions have not been completely understood. They are still matter of great research, but however we have identified one of the principal PAO, which is Accumulibacter phosphatus. So very relevantly named, and we have also managed to culture one of the PAO, but we still need to identify, and hopefully the fourth generation, the third generation sequencing techniques would throw a lot of light on PAOs. Alrighty. The next thing that is very important in built environment is our emerging contaminants. So the emerging contaminants, they are released in our environment, and in they are then microbes in the environment are exposed to them, and then they transform it, affect it, or degrade it, or just stay aloof to the emerging contaminant. Now as such, the emerging contaminants, they are continuously, I have mentioned this earlier in the lecture, they are continuously discharged and do not need to persist in environment to have an effect. For example, the birth control pill that is taken by women worldwide. Now this pill, when excreted through urine and released into wastewater treatment plants, it is not degraded in wastewater treatment plant, and when it reaches our water systems, it results in feminization of frogs and other aquatic lives, the male ones. And there is, the jury is still out that how it when this water is now taken up by drinking water treatment plant in the next city downstream, how it will affect the people. The presence of these endocrine disruptors will affect the health of people who drink this water. Now in wastewater treatment plant, how do we get rid of these emerging contaminants? We don't want that. We don't want these endocrine disruptors. Which might include pesticides or pharmaceuticals. We don't want them to last in wastewater treatment plant. So it is very important that we want to get rid of them in wastewater treatment plant. Many of these are xenobiotics, some are not, but many of them happen to be recalcitrant, they are not easily degraded. And even if they can be degraded easily, microbes don't really get an energetic kick or a lot of energetic profit out of them to want to degrade them readily. So what we often notice is co-metabolism. a microbe is trying to degrade another compound, it gives it a lot of energy, and the same enzymes, because they are functioning extracellularly, they also happen to degrade these endocrine disruptors, emerging contaminants. And this is an active area of current microbiological research. So again, for students who are very interested in research, this is very interesting, and I hope you look into it. All right. The next thing, when we talk about built environment, it is very important, is how do microbes or microbial activity destroy the built environment that we have made. Now, as human beings, we first focus on how they hurt us, and then we focus on how they help us. A good example would be pathogens were understood way before probiotic approach to treatment was understood, which is still being understood right now, at least scientifically. So, we are going to talk about microbially influenced or induced corrosion of metals, concrete and other surfaces. So, here you have a heavily corroded pipe, and this is a sulfate reducing bacteria. Now, if the water is carrying, if the pipe is carrying water such as marine salty water, or it is carrying sewage, then it will probably be very rich in sulfate, and then the sulfate reducing bacteria would thrive. They will use whatever organics are present in water, and they start reducing sulfate. Now, here is the thing, the sulfate that is reduced, when sulfate gets reduced, the FeO also gets reduced to ferric sulfide. So, this reaction results in destruction of iron pipes and causes serious corrosion. Now, recently it has been found that certain sulfate reducing microbes, they do not need to go through a long process to reduce the iron pipes and corrode them, but they can actually directly, the sulfate reducing microbes, they can directly receive electron from iron or give electron from the metal. Now, here is another picture where we are noticing the galvanic corrosion coupled with sulfate reduction by bacteria. So, here we have a sulfate reducing bacteria much like this one, and it is reducing sulfate into sulfide. The sulfide is forming iron sulfide, and also there is galvanic corrosion going on. So, we have anode and cathode, and then the Fe2 plus is converting into reduced FeS and releasing, some of them is just escaping into the water, some of them is getting reduced, and here we have a corrosion of the metal iron. And because of this corrosion, the way it works is that we have hydrogen present, and this hydrogen then encourages, the hydrogen is released, nascent hydrogen, which encourages the reduction of sulfate by sulfate reducing bacteria. So, all the three cycles here, the cathode, in galvanic corrosion, the cathode end reduction of hydrogen ion, the consumption of hydrogen ion to reduce sulfate ion quicker, and the using the end product of sulfate reduction for corroding the iron, all these three things are happening together and in good synergy with each other. The sulfate reduction of metal happens really quickly, and this has been a major challenge for many decades for communities that build, or for scientific community that and industrial community that is working on undersea pipes, undersea pillars, undersea posts. Okay. The next thing is, which is very important, is biodeterioration of stone and concrete. So, microbes do not only survive in sediment, in air, hardly any nutrient or very little, at least to human experience, very little mass to survive on, but they also survive in the rocks, in the stones and concrete. And this is a picture of biodeterioration of different concrete structures. So, you can notice there is some black thing, we often call it, oh, it is all moss or it is all algae, but it is usually a very diverse microbial community, and here we have algae and moss kind of microbial community growing. This is concrete degrading, and look here, is it not just concrete degradation, not just colonization of microbes, but the quality of your structure also degrades. Thus, we notice that concrete degrades. So, this is actually degrading the concrete and stone and not just colonizing it. Now, the microbes that live inside the rocks, inside the stones and inside the concrete, they are called as endolithic community. Now, this is a very classical picture that is used to give an example of endolithic communities that deteriorate stone. Here we have a very beautiful picture of a statue of an angel, and now after microbial degradation it looks unrecognizable nearly. Lot of it has degraded. Now, these microbes, they live in the small crevices and niches inside the in the stone and concrete structure, and thus they are called as endolithic microbial communities. Now, endo is inside, lithic is rock. So, the ones that live inside the rock. Now, because they are living inside the rock and there is not much nutrition available in the rock, they are called, this is then considered to be an oligotrophic environment for them. This is considered to be an oligotrophic environment, and their colonization of these structures depends on many factors such as surface roughness, the amount of pores that are available to it, the attachment surface that is available, and the mineral composition of the stone or concrete. Now, often what happens is again sulfate reduction and oxidation also at times, and that destroys the lime in the concrete and thus destroys the concrete structure. Another thing is, in case of fungal colonization of stone and concrete, the fungus will produce oxalic acid, which being acidic in nature does the same effect that acid rain would do. It destroys the surface of stone and concrete structures. Now, here is a list of different kinds of destruction that we can expect when it comes to biodeterioration of rocks. So, we can either have film formation or we can have and this film formation can happen on dense or fine grained stones, silicaceous sandstones, different kinds of stones, and then there is some information about the kind of microbial community and the kind of mineral there is. So, the kind of microflora, they are using a very nice term flora, because not all living members in this community which cause biodeterioration of stone and concrete are microbial. Some of them actually are visible like moss. They are usually superficial or inside the cracks. They are not found inside of nowhere in the concrete. So, on the surface, inside the cracks and fissures, and mostly unilamellar biofilm. So, they are not like water distribution system, where if this is your pipe, so you have biofilms going like this. So, it will not be like this, instead it would be very unilaminar. So, they would, why unilaminar? Because now they are exposed to different extreme conditions such as sun, wind, different changes in the humidity, desiccation or rain. So, because of that, any structure like this that wants to grow will be removed due to weather. Okay. Now, type of biodeterioration process would include discoloration, biofilm formation and local biocorrosion. We have talked about biofilm formation in water treatment, water distribution system. We have talked about corrosion briefly. And now, discoloration is that the biogenic pigments, they will oxidize iron or manganese and thus would change the color. The other kind of corrosion that happens is surface corrosion, and this happens to coarse grain. So, not fine grain will undergo film formation, coarse grain will undergo surface corrosion. In case of surface corrosion, often we notice that there is a deep penetration of the moisture goes deep, and thus the microbial community also goes deep up to 5 centimeter, and this is mostly bacteria. So, the biofilm is formed, this is extracellular polymeric substance that is released, and this actually allows microbes when they form these EPS biofilms in the rock pores, they actually increase the capillary water uptake. They result in biocorrosion by production of organic and inorganic acid due to microbial activities. So, dear students, now when it comes to built environment microbiology and how microbes destroy, very relevant to India is destruction of concrete sewer pipes, and actually elsewhere in the world too, because of biological activity. Now, remember, the wastewater that is carried in our sewer pipes is very rich in nutrients, very rich in carbon and biodegradable organics. So, now this wastewater undergoes very quick degradation of organics, and thus the conditions get anaerobic very quickly. In fact, when wastewater is collected, very soon you can see it turning into black because of production of sulphide. The sulphate is very quickly reduced to sulphide, and this changes the color of wastewater to black, and this is anaerobic condition. And when this sulphide interacts with water, it might produce H2S gas, and then this H2S gas is released up into the portion of the sewer pipe that has air. So, because sewer pipes are rarely completely filled, there is no pressurized collection system for sewer. So, the air component, when H2S is released, now there is oxygen present in the air. unlike what was in the liquid portion. And now in the oxygen component, what happens is the H2S gets oxidized. When H2S gets oxidized, what does it form? It forms H2SO4, which is highly acidic gas. It is very acidic, it creates very low pH, and that very quickly corrodes the surface of your pipes, and thus the surface of your concrete sewer pipes undergoes corrosion very quickly. And in fact, across the world, many concrete sewer pipes have collapsed and completely broken down because of this biodegradation based on first sulfate reduction in the water, and then its reoxidation once it goes into the air and formation of sulfuric acid. To prevent that, one of the things that people do is that they coat the concrete sewer pipes with special compounds to protect them from the corrosive effect of the very strong acid, sulfuric acid. The other thing that our marine businesses do is that they add nitrate. So they are the smart microbiologists. They will add nitrate. Now we know that nitrate is a better electron acceptor than sulfate. So instead of reducing the sulfate, microbes would like to reduce nitrate, and provided there is sufficient nitrate, they might not even ever get to reducing sulfate into sulfide until the wastewater reaches the wastewater treatment plant. And this way we can prolong the longevity of our concrete sewer carrying wastewater pipelines. Dear students, this is all for today. In the next lecture, we will go to other built environments and then move on to public health. Thank you very much.